Okay, folks, here we are. Going to do the second half of chapter 11. I've already put chapter 10, two parts there into your weeks 6 and 7, and here we have chapter 11. I've done chapter 11, put that in your pocket too, and here we are on page 393 of chapter 11. So we want to look at electrophysiology. When we think of that, we want to think about action potential. We want to think about nerve impulses, electrochemical signals. All those terms that I just mentioned really are the same thing. So as you look, on page 393, and you look in the second column, you'll see principles of electrophysiology. And as you look at the first few sentences, come down under the flashback there, and you see in muscle tissue, you are introduced to some of the concepts. Remember how you had positive charge on the outside of a cell membrane, and it was negative, or at least they showed it as negative on the inside of the cell membrane. It says it's the branch of physiology that studies electrical changes across the plasma membrane. So that's what we're really going to be talking about today. We're not going to get too deep into it, but it's a little tricky. You have to read over it several times, kind of get it cemented in your mind there. So there can be electrical changes across the plasma membrane. You saw that in the muscle, where the sodium ions rushed in and turned the inside of the membrane positive and the outside was negative and so forth. So that's what we're really talking about. But now we're talking about it in terms of neurons. So you can go back on page 354, I think it is, and you'll see those terms we talked about a little bit, called resting membrane potential and so forth. So let's look down in this second column on 393. You see the title, Ion Channels. Remember that? See, this goes all the way back to chapter 3, where you had those proteins floating amongst the phospholipid molecules. And those proteins played a role in allowing things to come in and go out of the cell. Some of those proteins were actually pumps that pumped ions out or in or whatever. So we want to look at those proteins in the cell membrane and remember what they do. You can't live without having some charges in your neurons and charges going down your muscle, sarcolemma, and so forth. So look over on page 394. So we got that straight. On page 394, and you come down to the bottom of the first column, and you see again it says you've been introduced to vitally important concentration gradients. That was back in the first chapter. Gradients, go look that up. Make sure you understand what that is. If you don't have gradients, then we're dead in the water. That's it. So you got to have these gradients in terms of these ions in order to have the body function. So as you see there, it says you've been introduced to vitally important concentration gradients of sodium and potassium that exist on either side of the plasma membrane. And you see as you come down, it says in a neuron, it's about 5 or 6 lines down, 6, 7, 8 lines down. As in a muscle fiber, the concentration of sodium ions is higher outside, that's in the extracellular fluid, than it is in the cytoplasm. So the sodium concentration is higher outside than the inside. Potassium on the inside is higher than it is on the outside. So you come down to thinking about concentrations, and since these are charged ions, you got electrochemical gradient there. It's a chemical. These are chemicals, sodium and potassium, but they're also charged. they've lost an electron, so they're a cation. And so we can talk in terms of electrochemical activity. Now, look at the last little paragraph. These gradients are maintained to some degree, even created by the sodium-potassium pump. Now, that's a protein, believe it or not. It's a pump. It doesn't contract. It doesn't carry substances throughout your bloodstream and so forth. It's part of the cell membrane, and it acts as a pump. Kicks the sodium out, brings the potassium in. But there's more sodium out there than there is potassium, and so we've got a polarized membrane. Polarized means you've got certain charges up here, and you got different charges underneath, or within, I should say, charges that are different from the charges that are inside a cell. So as you look at the bottom of the 394 in the second column, you see resting membrane potential. We've already talked about that back in chapter 10. So these proteins create this polarity. Some people call it polarity. That just means you got different charges because you got different numbers of ions on either side of the plasma membrane. Some people want to call it a membrane potential, and that's okay. Same thing. When you've got resting membrane potential, some on the outside, some on the inside, then you have that difference, which they call a potential. And a potential means that you can have the possibility of doing work, accomplishing something with those ions being different. If they were the same number on the outside as they were on the inside, it's called dead. It's called dead, okay? And they talk about voltage. That's just the separation of the charges. 15 out here and 5 in here, something like that. So anyway, I want you to look at the top of page 395 and look at that first column. And you see the illustration there. It talks about minus 70 millivolts. That's the voltage across the plasma membrane. It's a minus number at this point. It's positive on the outside, negative on the inside. And we're not going to get into why it's all positive and negative, but you just want to understand it is. So it's more negative on the inside than it is on the outside. And as you read through this, you'll see the word. It says the negative voltage is present. The cell is at rest. It's the resting membrane potential. And it says that's why they call it that. And the cell is in the state of being polarized. Your battery in your car is polarized. It's ready to do some work for you. There is a potential for work there. Now, it doesn't shoot ions out like we're talking about here, sodium and potassium. It shoots electrons down wires that go to your starter that get turned over, and next thing you know, your engine's running, and so you're happy and you're ready to go to wherever, Harris Theater, Food Lion, get you some food or whatever. So you can see in the resting state, in the case of neurons, which is what we're talking about now, you've got a positive charge on the outside, just on the outside of the membrane, not way on away from the cell membrane. You got a negative charge on the inside. At least that's the way they've chosen to get you to picture differences of concentrations and therefore of ions and therefore differences in polarity. Now, let's go down to 395 and you see electrochemical gradients. And we mentioned that just a few minutes ago. See, chapter 1 is going to be coming right up into this chapter. That foundational material is important. If you don't understand it, go back and read it. Okay. So you look at the bottom of the second column and you see electrochemical gradients. Let's flip over to page 396. And we've already mentioned that term, electrochemical. That's your sodium and your potassium. And electro tells you you've got charges, different charges on each side of the membrane. And as you read this first little paragraph, it says, we've all seen how solids move across membranes by diffusion according to their concentration gradient. There's more sodium outside than there is inside. There's more potassium inside than there is outside, but they don't equal each other. If they equal, remember where you are? You're going to be planted. You're dead. So it says, in fact, the concentration gradient is the main factor. The concentration gradient. There's more sodium outside than there is inside. We have a gradient. Determines the movement of the uncharged molecules, solids. Well, it's going to do that same thing with the charged ions. And so you come down to the last couple of sentences. For this reason, diffusion of an ion across a plasma membrane is determined by both its concentration gradient and its electrical gradient. Positives are always going to move to the negative area and so forth. So that's electrical. So we have an electrochemical gradient on the plasma membrane. Now, as you look at the bottom of page 396, second column, you see it says changes in the membrane potential ion movements. When the ions, like sodium and potassium, start moving, that's what creates an electrochemical message or signal or action potential or nerve impulse. All those terms are basically the same thing. So let's look over on the first column on 397, and you're in the first paragraph. So come down 1, 2, 3. Well, let's just do the first couple of sentences. The resting membrane potential comes into existence from unequal distribution of ions. Now remember, ions are charged, so you've got unequal distribution of charges, charged molecules, and their different abilities for crossing the membrane. If we change the ability of the ions to cross the membrane potential will change. That's how we start to generate a nerve impulse, by moving these ions. That impulse is due to the moving of ions. The reason my hands are moving and my lips are moving and so forth is ions are flowing back and forth, and they create nerve messages, so to speak. And of course, you got to recharge that nerve, that neuron, and you've got those pumps that do that. It's just amazing to think about what is going on inside of our bodies, and we just take it for granted. All we want to do is, well, let's go watch a good movie or get some food or something like that. So you come into where you see figure 11.14a. Now don't go look down there. I'm not interested in you looking down there right now. You see it says, as shown in that figure, if gated sodium channels open, tells you they were closed, right, at first, but now if they open, sodium ions follow their electrochemical gradient. Positives go toward the negative. The greater number of sodium outside the cell membrane wants to get inside where there are fewer sodium molecules, sodium ions. It's all about trying to balance out. But if it balances out, we're dead in the water. That's why we've got these sodium potassium pumps that restore the polarity and keep it there so we're ready to talk and listen and take notes and so forth. So you see it says the influx of positive charges makes the membrane potential more positive. That influx of the sodium ions coming into the cytoplasm is called depolarization. That's your definition for that. And you see it says by this process, the cell becomes less polarized. If all those sodium ions are moving in, you can imagine there's a point where everything's kind of equal with sodium. But it keeps moving in to some degree, and next thing you know, you got an unbalanced lining. It goes positive on the inside, negative on the outside. By this process, the cell becomes less polarized as its membrane potential approaches zero. When a cell returns to its resting membrane potential, then the process called repolarization has occurred. So it's like it fires, and then it's got to be charged again. The firing comes about by depolarization. The recharge to get ready for another event is called repolarization. And in the middle of that is your message, called an action potential, that travels from here down to my mouth and helps control my tongue and lips and so forth as I try to get this information somewhat understandable to you. All right, let's go over to page 398. Page 398. And you look at the first column that says action potential. Action potentials, remember, are the electrochemical signals. They're also called nerve impulses. So that's the movement of ions. So here's your definition of an action potential: a uniform, rapid depolarization and repolarization of the membrane potential of a cell. Yeah. Come on down a couple of lines, and it says within the nervous system, signals are sent through an axon to another neuron, a muscle fiber, or a gland, and you get a result. In my case now, my muscles are contracting in my fingers and my mouth and so forth. The head turns this way and that way because action potentials are going to muscles. Excuse me. They're going to muscles and they're causing contraction, like we studied in chapter 10. So we're tying that in with chapter 10 because we did muscles first. Now, I want you to look on 399 and you see a little sequence of an action potential being formed, of depolarization, of repolarization. So as you look at the first part, you see most of that cell membrane has positives on the outside, negatives on the inside. Sodium's on the outside, potassium's on the inside, and you got an unbalanced number of those, and so you have a polarity where the outside is positive and the inside is negative. It's really less positive, but they choose to use the word negative. Makes it easy, I guess, to understand. So when some sort of stimulus occurs, for instance, I got this rash on my hand here, and it itches. It's probably poison ivy or something. I was cleaning out some stuff in a garden. Well, it's itching. Now, that obviously is not going to keep me from teaching you or getting home or driving down for supper or something like that. But the point is, I'm aware of it, see? There's an irritation there. And I term it as itching. So what that irritation does is it makes itself known to me through this action potential that will pass down the membrane of a nerve from my hand to up here, and I'm aware what is going on the back of my hand. And I want to scratch it. And of course, it feels good to scratch. You can't scratch it too much, you mess up your skin. But when that message, when this itching creates this little message, you see where it says on the left-hand side is local potential? Notice it's positive on the inside and negative on the outside. That means that a sodium channel has opened up and sodium has rushed in, and that's the start of a nerve impulse to tell me, you got poison ivy. You got some little blisters on the back of your hand, and they itch. Okay? And so this is a step-by-step process. If you move to the right, you see it's still positive over there, on the outside and negative on the inside, but come down to the next illustration and you see negative on the outside and positive on the inside. Okay, that's the impulse moving. That's a reversal of polarity. It's a reversal of potential. Now, as that negative area, that outside negative and that positive moves to the right, look what happens in the next one. You see it's positive on the outside, negative on the inside. That's repolarization. And so the nerve is ready to fire again and send me another message. We keep sending the messages all day. Your hand is itching. Your hand is itching. You know, you got an irritation there. Oil from the poison ivy vine. And so you can see that eventually the whole cell membrane gets repolarized. See how potassium, look in the third part. Look at the third illustration where potassium comes rolling out. Now you got some positive ions outside. You see in the illustration above, sodium came roaring in. It turned negative on the outside, positive on the inside. That's your message. But then pretty soon this protein channel kicks out potassium, and so you start getting positive ions on the outside and restore the negative on the inside. So you're back to the membrane potential, the resting membrane potential. So if you can understand that you're moving positive ions in and out, and that changes the polarity, that's the message. Isn't that amazing? And all I do is say, oh, I got something on the back of my hand. I got these little blisters. Need to put some cortisone on it so it'll stop itching. It kind of soothes it. So I guess this doesn't occur. All right, so that's about an action potential there. Now let's look over on page 402. Now here they do the same thing we were looking at on the previous illustration. We're looking at figure 1118. And so you can see the steps they've got, the events of propagation or keeping the action potential going. You see it says the axolemma. What is the axolemma? It's the plasma membrane of a neuron, isn't it? Just like sarcolemma is the plasma membrane of a muscle cell. Axolemma depolarizes. Don't worry about threshold and all that, but it depolarizes. That's the moving of the sodium into the cell. That's the start of the action potential, but it's also called depolarization. You can see it in the first illustration, page 402, come down to see number 1. See how the reversal of charges has occurred? That's the start of your action potential. It's the result of the sodium moving in. That's depolarization. Look at the rest of the axon there. It's all positive on the outside, negative on the inside. It's got to move all the way down. And so you look at the next picture, and you see the little green area, depolarizing membrane, action potential, negative on the outside, positive on the inside. But look to the left of that area where the action potential is. It's back to positive on the outside, negative on the inside, ready to fire again. So you can picture that, those ions changing, and depolarization is occurring and repolarization is occurring right behind it. So when the message gets to my brain, there's another action potential coming. Mr. John, your hand is itching. That's what happens. Movement of sodium and potassium ions. And you got proteins in that membrane that will restore the resting membrane potential. It'll eventually kick the sodium out and bring the potassium in. And so we get that concentration of potassium on the inside and sodium on the outside. We're not going to get into all the depth of that, but hopefully you have some understanding of that. Now, there are, if you remember correctly, neurons that have no myelin. Remember, myelin was a lipid material. and it acts like insulation. Insulation's good. You want insulation on the wires in your wall. You don't lose electrons that way and create fires and so forth. It's good to have that insulation. So you remember some of the cells, the neuroglia, I think that's what the book wants to say. They will surround axons with their body, and that becomes a wrapping and an insulation around the axon. And you remember there were little breaks. Look on page 403. And you have a contrast here. At the bottom, you see pictures of a myelinated axon versus an unmyelinated axon. Now, what I want you to do here is just get sort of an idea about this saltatory conduction, which is very fast, very quick, under conduction speed. And you see the bottom of that little paragraph, saltatory conduction. So what that means in a myelinated axon is you look down, only the nodes discharge, get polarized and repolarized. It happens very quickly. It's not the easiest thing to understand, but what I want you to just take and believe and apply is that where the nodes of Ranvier are located, that's where depolarization and repolarization occurs. Even the diagram is a little bit different down here. But it's sort of challenging to try to understand that. But this is what I want you to know, just so you can answer the questions and understand that with only the nodes depolarizing, you don't have to redo the whole axon. Okay, that's what I want you to remember, and it's very quick. So it like jumps from node to node. That's saltatory conduction in a myelinated axon. Otherwise, you have to do the whole neuron. All right, let's look at page 406. And we're talking about module 11.4. You see it says neuronal synapses. Come down to the bottom and you see there is this way out. Okay. At the bottom of that first column, you see the word synapse, and that is the junction of two neurons. It can also be the junction of a muscle and a neuron, but here we're talking about neurons, neuronal synapses. And so since you've got, let's say this arm is a neuron, and this in the fist, that'd be the bulb. Remember the synaptic bulb, telodendria? Here's another one. They're like this. They're very close to each other. And so this one, if it comes, say, from my sending a message to my arm, this would be a presynaptic neuron. This would be a postsynaptic neuron, because this message is coming this way and it's got to jump over into the next neuron. So we have continuous information or signals passing to parts of the body. So you look in the second column and you see the words presynaptic neuron and postsynaptic neuron. The one before the synapse is called the presynaptic and the other one's called the postsynaptic. Now we've got a couple of kinds of synapses, and you'll appreciate something when you hear you've heard it before. This comes from like let's see, maybe chapter 4 or something like that. Look on page 407 and you see electrical synapses. Occur between cells that are electrically coupled through gap junctions. Now, in this Word document that I'm going to send you, that I've already sent you, I think, I've already mentioned in there, do you remember that? Yeah, see, this stuff comes back. That's why I tell you, read it over, read over the old material, because we're building on the old material. I say the old, the first few chapters or so of the book. So anyway, notice as you read through there, it says that the gap junctions contain precisely aligned channels that form pores through which ions and other small substances can flow. Ions, action potentials, communication, results through that communication. So that's an electrical synapse that you see down at the bottom, figure 1122, and you see in part A. You've got those gap junctions so that ions can pass from one cell to another. Now, the other type of synapse is called a chemical synapse. It still involves electricity, but it's also involving a chemical that we call a neurotransmitter. So, as you look to your right, there's an illustration of a chemical synapsis, or a synapse, I should say. And you see the presynaptic neuron, axon terminal. They don't describe the synaptic bulb, but that's that enlarged area. And you see a synaptic vessel in there. You see neurotransmitters. That's a chemical in there. Remember how the vesicles had to fuse with the axonema? And then it dumped in that substance acetylcholine to attach to the proteins in the sarcolemma so that sodium could come in through open channels. Without that acetylcholine, that would not happen. There's your chemical, the neurotransmitter. So really, this deals a little bit with electricity, and then you also have a chemical that enables the message to move from one neuron to another. That myoneural junction, or neuromuscular junction, was a chemical synapse. It's really electrochemical because you got the action potential that comes down. So as you look at this, you see it says the vast majority of synapses, this is on page 407, second column, under chemical synapses. More common, and you got many more of those. They are more efficient, as you see there. And let's see if it needs to say anything else. Notice a couple sentences down, it says converts an electrical signal into a controlled chemical signal. So that's why they call it a chemical synapse. So there is no loss of strength. The chemical signal is reconverted into an electrical signal in the postsynaptic neuron. So you got a neuron here, got a neuron here, got a action potential, which is an electrical thing. It's ions and so forth, and then it causes the vesicles in the bulb to fuse with the cell membrane. Out comes the neurotransmitter, which, as you see, is going to touch the little purple things. Remember, that's very similar to what we had with the muscle. Those are sodium channels. They don't open unless an acetylcholine, in this case, the neurotransmitter, attaches to it and opens them. Boy, it's amazing this body works, isn't it? Fascinating. Now, one thing they left out in the neuromuscular junction was this. Look on page 408. And you see figure 1123. And there again, you see the presynaptic neuron and the postsynaptic neuron. And you see the action potential, that's a little green arrow that's coming down. They left out, though, is you see calcium channels. The calcium is in the extracellular material, and these channels here, when that action potential comes down, when that electrochemical signal comes down, it opens the channel of the calcium channels, and calcium comes into the presynaptic neuron and causes the synaptic vesicles full of neurotransmitters to fuse with the cell membrane of this presynaptic bulb. Did you get all that? It looks so cumbersome, doesn't it? But just look at the Olympics. Go to look at the diving that they do and so forth, and the running. It happens very quickly. Very quickly. And so you can see that the little vesicles fuse with the end of the synaptic bulb, and they dump the acetylcholine, in this case, into the cleft, synaptic cleft, that then connects with those sodium channels, opens them up, sodium comes in. Now you got an chemical signal, you got an electrochemical signal, the action potential going down the other neuron. It's pretty nice over here on page 409. Now you want to be able to identify those structures on that, understanding the synapse. You look on 409, first column, and you see the first column 1, 2, 3, and 4 steps. That's a pretty nice way to put it. So you want to know that. It might be one of those questions at the end. Explain the transmission of a message from one neuron to another. Now, when that transmission occurs, you got to stop it, and you got to start it over again. It's not like it just flows and flows and flows. It does so very quickly. It might seem like that, but that doesn't happen. Look on 410, and you see termination of synaptic transmission. Now, there's several ways that can occur. We're just going to learn one. As you come down, you see these bold print terms like diffusion and absorption, degradation in the synaptic cleft, and retake up of the presynaptic into the presynaptic neuron. What we're going to do is learn that middle one. Degradation in the synaptic cleft. Now, if you degrade something, then you break it down, don't you? And so what you see here is it says certain neurotransmitters, not all, but certain, broken down by enzymes. Go back to chapter 10. You remember the neurotransmitter that enabled the action potential to hop over the little cleft, the little space, into the sarcolemma and get into that muscle cell? That was acetylcholine. And it produces also, they also have the ability to produce an enzyme. In this case, it's acetylcholinesterase. That's a good one to get your mouth and tongue working at 6 o'clock in the morning, isn't it? And the acetylcholinesterase breaks it apart. It's reabsorbed into the presynaptic neuron and reconnected. So you use it over and over and over again. Kind of like plastic, isn't it? We drink out of it, then we throw it away. Next thing you know, somebody's made a shirt out of it or a sweater. That's fascinating. But anyway, get the point. Got to stop the transmission. Now, I want you to read, as you look at page 410, look at the second column. Because this is a possibility for some of us. We have garages and you have things stored. And I remember my son had a box of those quart-size containers for oil for his car. And he was pulling them out. One of the time they'd been open and stuffed aside for a while. And he was putting oil in his car and so forth. And I happened to look up under there, shine a light in that bucket. There's a black widow in there. Read that about black widows. You don't want to get bit by them. You're not going to die, most likely. but it's not going to be any fun. It's not like poison ivy, okay? So read that. Might have a question or two on that. Got a nice picture over here on page 411. Shows that action potential coming down. Number 1 there. Number 2, synaptic transmission occurs. It's giving you these nice steps. Don't worry about number 3. We're not going to get into those postsynaptic potentials. But then the termination of synaptic transmission down at the bottom. Okay. Oh boy, we need to close it up here for a second. Let's go to page 413. And you see it says in module 11.5, page 413, about neurotransmitters. We mentioned acetylcholine. Look on page 415. See acetylcholine in the first column. The synaptic junctions, the synapses that use acetylcholine are called cholinergic synapses. And of course, that would include the junction of the nerve and the muscle, neural junction. You come on down and read a little bit more, and you see acetylcholinesterase. And so that's how that's the enzyme that breaks it down into acetic acid and choline. Okay. So that's a neurotransmitter. You've got a chart down here, but look on 415 at the second column, and you see biogenic amines. They're different. And you know some of the names of them. Just some examples here: norepinephrine. Norepinephrine, widely used by the ANS. You know what the ANS is? That's the autonomic nervous system. That's one that we don't have any control over. Heart rate, blood pressure, digestion, and so forth. And you come over to page 416, and you see epinephrine, which acts in a very similar way to norepinephrine. Says it's used more as a hormone by the endocrine system, which we haven't covered yet. But you've got a couple of glands on your kidneys called the adrenal glands, and that's where epinephrine is produced, called adrenaline also. And it gives us that ability to deal with a situation, whether we need to flee or we need to fight. Then dopamine, as you read through that, you see it's involved in emotion and motivation. So again, you got to have the right amount in there. If you don't have enough, you're just depressed. If you got too much, then you're wiggling all over the place and shaking. So anyway, and those receptors for dopamine in our central nervous system, that's where people sniff cocaine and take amphetamines. They try and get high. So not a good way to spend your life. Just need some help, ask the people that will help you. Serotonin, come down. It's not in bold print, but it's in the next paragraph. Serotonin comes from the amino acid tryptophan. And they're not totally sure about it, but it seems to be involved in maybe depression, things like that, mood regulation type stuff. Then you see histamine in the next paragraph, and some of you have probably taken antihistamines. Antihistamines will open your respiratory tract. When you've got a cold or whatever, histamine is involved in inflammation. And so you take an antihistamine to kind of calm that inflammation down so you can breathe at night. But anyway, that's a neurotransmitter. Comes from the amino acid histidine. And that is the end of chapter 11. All right. I'll probably be able to put this into your D2L tomorrow. I can't seem to get it right after I made it. It's got to sit in there and age like good wine, I guess. Anyway, I'll put that in your D2L packet tomorrow. Okay? All right. So you've got the 2 Word documents now, and then this will be the last video on chapter 11. So use your time wisely, please. I'll see you later. Bye.